All right. So we're going to talk briefly about different treeing algorithms as well. We talked about neighbor joining. Um, this is a really simple algorithm. It's really fast. I can run 1,000 neighbor joining trees on my laptop in something like 20 seconds with a good size alignment. And it's useful for that reason. It also turns out that for ribosomal sequences, it's a pretty good approach. It gives you pretty robust, pretty useful trees. Trees that turn out to be consistent with other good tree algorithms. Another way is a program called an approach called Fitch-Margolius. The major difference here, it's also a least squares distance matrix method. So it starts out by taking the alignment and converting it into a distance matrix. But then instead of separating the process of tree structure and branch length, which is what neighbor joining does, you solve the two simultaneously. But instead of in order to separate the work, what you're separating out is the sequences themselves. And so you start out by doing both the distance and structure of just two sequences, and then you add a third sequence and a fourth sequence, and you keep adding sequences to the tree, one after the other. So you're solving for structure and distance at the same time, but feeding the tree one sequence at a time. You're building the tree one sequence at a time. An easy way to think about this, in this tree, you've got all these evolutionary distances in a table. Think of them, if you're adding a sequence, think of that in 3 dimensions. This tree is in 2 dimensions. Now imagine you've got a sequence out here connected to each of these other ones by a string, the length of the evolutionary distance. And then to add it to those 2 dimensions, you just push it down and run the string along the lines that it fits with until they all converge together and you create a separate branch. That's a kind of visualization of how Fitch works. When you're doing Fitch trees, one of the things you worry about is how the order of sequences you add can affect the trees you get at the end. And so very often what you do is a bunch of trees in which you randomize the order that you add sequences in and see how they compare. And if it's a good tree, they ought to be pretty similar, right? Another approach is parsimony. This is based on Occam's razor. So the notion that the tree that demands the fewest changes in the sequences in evolutionary history is the most likely tree to be correct. And so if you've got 4 sequences like so, you can go back and identify 2 similar ones and what sequence it would have had as an ancestor, and go back, and you're actually calculating the ancestral sequences as you go along. The problem with this is that it is the math of this falls into a category of mathematical problems called NP-complete. Any of you ever heard of that before? Basically what it means is, although there's only a finite number of possible trees to search through, that number is very large, astronomically large, far more than a computer could ever search through. And so you have to take shortcuts as you go through this process, searching through all possible trees. You might imagine that you could create a collection of all possible trees for sequence alignment, and then sift through them and find the one that has the smallest number of sequences. But you can't logistically do that. So you have search patterns and so forth you use in order to do this. A byproduct of this is these ancestral sequences. This can be useful at times. I think it was Brenner who did this cool experiment looking at lysozymes. Your body has a bunch of lysozymes in it, right? There's one in your eye that keeps bacteria from growing in your tears. There's one in cows. There's one in the gut. It's expressed in the rumen. In many animals, there's one in the oviducts. Eggs are full of lysozyme. So what was the function of the ancestral lysozyme? Well, he went back, he made a parsimony tree of lysozyme, and then he made that protein and tested its properties. And it turns out to be an oviduct lysozyme. So that's it. And so the lysozyme in your eyes evolved from a gene duplication, starting with an oviduct lysozyme. And likewise in rumens, the common ancestor of rumens duplicated the gene and took the oviduct lysozyme and turned it in that case, the I lysozyme, and turned it into a rumen lysozyme. The reality is that for ribosomal analysis, and in fact RNA alignments in general, parsimony methods, and there's a whole bunch of different approaches to doing parsimony, don't in general give you as robust results as other approaches. And so you don't see a lot of people rely primarily on parsimony methods, but maybe use it to confirm something along those lines. One of the best approaches to the analysis of alignments to creating trees is a maximum likelihood method. This is a heuristic approach. So you're calculating the parameters that you're using for your substitution model at the same time you generate the tree. You start out with random numbers for substitution model parameters. You start out with a random tree. And then you make a change and calculate the value of the tree. And if it's an improvement, you probably keep it. And if it's worse, you throw it away and try again. There's a random number associated with that, so there's only a probability that you'll keep it or a probability that you'll throw it away. And every time you tweak, you randomly change one of the substitution parameters and the tree structure or branch length, and then decide whether it's an improvement or not, and can repeat that over and over and over and over and over and over again, until your trees aren't getting any better, no matter how many times you try to tweak them, and you consider yourself done. This works out really well. This is probably the best, most well-accepted approach to generating phylogenetic trees, both from RNA and protein sequence alignments. Yep. Typically, instead of starting entirely random, it'll run a neighborjoining tree and start with that. You go back to a standard least squares neighborjoining tree, and whatever tree you get, which is probably pretty good, is your starting point. You don't have to do that, but pragmatically, it takes a long time for the analysis to get close enough to the correct tree for it to start to slide down the slope toward optimizing the tree. So usually a guide tree shortens the calculation time dramatically. And this is the problem with maximum likelihood, is that it is excruciatingly computationally intensive, particularly as the alignments get long and as the number of sequences gets large. With an alignment that I can run 1,000 neighborjoining trees in a few seconds, might take me a weekend to run one tree with neighborjoining. I can't tell you how many times I've sat there with my laptop running for 3 eggs on one tree. And these things are fast, right? And that's assuming you take some shortcuts, like starting with a guide tree. There's some other kinds of kind of shortcuts that usually turn out better and dramatically cut the testing time, but can sometimes go haywire. The last one that people are using more and more, but still hasn't really gained a lot of acceptance, are Bayesian approaches. It's a little bit like maximum likelihood, but it uses Bayesian math to generate a tree. I really don't want to get into it. The biggest issue is that Bayesian trees can get trapped in kind of a local optimum that isn't correct, and it's hard to get them out of that. So let me see. There are lots of treeing algorithms. I think the next one is something else. Yeah. So there are lots of treeing algorithms. I will say that the algorithm you use to calculate the tree is one of the least important issues about how you do the analysis. If you've got a good alignment, any of these algorithms is going to give you a reasonable tree. That's why I like Neighborjoining, because it's so fast. And I'll show you why that's important in just a minute. Normally, people will generate trees using two fundamentally different approaches and compare them. Because if a tree is robust, if an alignment is good, you ought to get pretty much the same tree either way, right? Because it's the real tree. For ribosomal sequences and for RNA sequences in general, Neighborjoining and maximum likelihood are kind of the accepted standards. For protein sequences, particularly when they're closely related, parsimony methods are great.